everyone. Today we are going to talk about the second chapter, Sampling and Pretreatment of Analytical Sample. This chapter mainly consists of two parts. 1. Sampling and preparation of analytical samples. 2. Pretreatment of samples. In the last lecture of Introduction, we learned that analytical chemistry mainly focuses on quantitative analysis, which usually consists of the following 5 steps. 1. The collection and preparation of samples. 2. Sample pretreatment. 3. Masking and the separation of interfering components. 4. Measure. 5. Result calculation and evaluation. In these 5 steps, sample collection and preparation are the first problems encountered in the analysis of complex samples. In a quantitative analysis, the amount of measured sample usually is sub-gram to a few grams, and the analysis results of which will be taken as the actual condition of several tons or even tens of millions of tons of materials. So the average composition of the analysis sample must be able to represent the average composition of all the materials. Otherwise, the analysis results will be meaningless no matter how accurate they are. Therefore, it is crucial to collect and prepare the proper sample according to the specific situation before determination. So, how do we collect an analytical sample? First, we will study the collection and preparation of solid samples. Let's take ore, soil, and metals as 3 examples. Collection of samples must comply with 4 principles. 1. Survey and collect data on site. 2. Make sure the representative sample is collected. 3. Meet the requirements of sample amount. 4. Store samples in correct methods. The key of these 4 principles is that the sample should be representative. The composition of collected sample must be able to faithfully represent the bulk of materials. Otherwise, the analysis results are meaningless, even if they are accurate. Now, we will take a look at some notes before collecting all samples. First, arrangement of sampling points. All samples are usually collected in cars or train carriages. Depending on practical situations, like the material stack depth and location, the distribution of sampling points should be reasonable and homogeneous. 4 different schemes for setting sampling points are shown in Figure 2.1a, b, c, and d. Second, because the porous surface of solid sample absorbs water in the air, the solid sample always contains moist water, also called hygroscopic water. This water needs to be removed before the sample analysis. The purpose of dehumidification is to compare the quality fraction of each component. The methods of dehumidifying, including drying in oven, spreading on the ground for natural dry, or drying in a vacuum drying oven. Then we can prepare the ore samples. The larger the ore size is, the more sample needs to be collected. It is required to be broken, sifted, mixed, and reduced in 4 steps to prepare a small amount and highly uniform analysis sample from the original ore bulk. After rough crushing, the ore samples are screened by number 4 to 6 sieve, a certain amount from which was collected for further crushing. The obtained sample is sifted by number 20 sieve, a reduced amount of which will continue to be ground. The commonly used method for reduction and division of sample is called the cautery method, which is to mix the comminuted sample uniformly and pile up the sample into a coal and then flatten. It is divided into 4 parts through the center, 2 parts of which are discarded randomly while rest 2 parts are collected. Thus, the sample amount is cut in half. Next, we will study the sampling of soil samples. First, set the sampling points. On the basis of investigation and research, a certain number of lands which can represent the research regions are selected as sampling units, and a certain number of sampling points are set in each sampling unit. In each sampling unit, multi-point sampling should be carried out in different directions, and the collected soil samples are mixed uniformly to be representative. Depth of soil sampling is decided according to the modeling purpose. If only the condition of soil pollution is investigated, it is generally necessary to take the surface layer of 0 to 15 cm or 0 to 20 cm, which is a ploughing layer, and use a soil shelf to sampling. If we want to know the depths of soil pollution, stratified sampling should be carried out according to the soil profile shown in the right image. Sampling time of soil samples should also be considered. Samples can be collected at any time if we just need to determine the condition of soil contamination. If we need to master the contamination of crops' growth on the soil, samples should be collected according to season changes or crop harvest. Amount of soil sample. Generally, 1 kilogram is required. Soil samples are normally the mixture of samples collected from different sampling points with large quantity. Common quantitative analysis just needs 1 to 2 kilograms, so the mixture amount is reduced by quartile method repeatedly to get the final soil sample. Preservation. The preservation containers of soil samples include glass bottles, polyethylene plastic bottles, and cloth bags. Keep samples away from light in dry and clean places. If the components we determine are labile components such as volatile phenol, ammonia, nitrogen, and cyanide, fresh soil is needed. The determination of most other components requires dry soil samples. Samples of matter or metal products. The metal samples are homogeneous because of the high temperatures melting. Therefore, the cutting part can be used for quantitative analysis for flake or wire samples. For steel and cast iron, because the solidification temperatures of iron and impurities are different as well as the different solidification time at surface and internal, composition of the sample surface and internal is uneven. So we should first clean up the surface first, and then drill to get the clastic samples at different position and different depths, which are finally mixed evenly as the analysis sample. These are the sampling methods for 3 kinds of solid sample: minerals, soil, and metal samples. We have 2 questions for you. What is the principle of sampling? In order to understand the effect of phosphorus fertilizer on rice growth, a small shelf of sample is taken from a rice field. Is the analysis result of the collected sample meaningful? Why? That's all for today. Thank you.